Okay? And this will eventually lead us into DNA packaging. We'll talk about the histone code. We'll talk about gene regulation a little bit. So, simplest idea, simplest way of packaging genomes. So, DNA is this long, thin molecule which can float around. Now, again, it's got the sugar phosphate backbone. There's negative charges spaced every so often, right? Spaced equidistant from each other, right? The negative charges do not want to be hanging out, so they try to stretch the DNA out as far as they can. But if you put it in an aqueous solution, it's waving all around like this, right? And you get to a couple thousand base pairs, and there's enough flapping around with the DNA that it could actually sort of bounce back on itself like that, right? So DNA, nucleic acids, if they're going to exist in an organism, if you have a whole bunch of DNA, like the size of the human genome, to compact that all into this tiny little space of a nucleus, the DNA somehow needs to be grabbed and brought and held together and compacted in some manner. And people thought that it was just kind of held together. There were proteins that randomly held it together, and we now know very different. DNA is organized in a very, very specific manner. The more I learn about the cells, the less I believe anything random happens in cells. Okay? So the simplest way that something could be packaged is how a virus packages its genome. So this is a this is probably tobacco mosaic virus, and it forms basically this long cylinder-shaped structure. The genome happens to be RNA, and the RNA kind of coils around, and proteins grab onto it. The proteins have a lot of positive charges. The positive charges interact with and counter the negative charges of the genome, and then the proteins kind of hold on to each other to form this basically protective shell. And you probably learned this in General Biology 1 or General Biology 2 as the capsid, with a structure that protects the genome from getting degraded. There's a very simple way of packaging the genome. In bacteria, they package their DNA what we call a nucleoid region. So when I was in your shoes, we were not aware of this. People were starting to propose it, suggesting that bacteria took their genome and put it in a specific spot in the cell. And other people said, no, the genome is just kind of hanging out in the cytoplasm, and it occupies the whole cytoplasm, just this huge meshwork throughout the whole cytoplasm. Pardon me. That's not necessarily the case. What someone has done here in this image is there's a chemical called ethidium bromide. And that chemical intercalates into DNA. So if we have one strand, 5 to 3 this way, the other strand goes 5 to 3 this way, right? And we have the base pairs running like this. Ethidium bromide is a ringed structure chemical. It's flat like this. And I think there's 3 rings to it. And I don't know the exact structure of it, something like that. There's a bromide hanging off on it like this, right? This chemical prefers, instead of being in an aqueous environment, it actually prefers to slip its way into this little space here. And ethidium bromide is able to absorb, every chemical can absorb certain wavelengths of light, right? There's a spectrum. Every chemical has a spectrum that it absorbs. That chemical absorbs in the ultraviolet, and that energy, those photons, or those waves that get absorbed, they kind of reverberate around the ring structures and eventually get emitted at a lower energy level, so that the light gets emitted in a visible wavelength. So what someone has done is they've taken bacterial cells and they've dripped a small amount of ethidium bromide into solution. The ethidium bromide prefers to be in this environment than in solution, and so it concentrates in the DNA, intercalated into the DNA. The word is intercalated. Into the DNA like that. And so anywhere that there's genome, anywhere that there's DNA, this organism will light up if you shine UV light on it. So here's a cell which looks like it's just about to divide. The chromosomes have separated to opposite sides of the cell. In the cell wall, it looks like the organism maybe already has pinched closed here. So here's the genome and here's the genome. Here's another organism which looks like it's just starting to divide. Right? So we can see where the genome is by labeling these things. If we actually look at the genome of a bacterium, did you know that's a bacterial cell is a bacterium? When you say the word bacteria, you're talking about a population of things. How did you know that? If we look at the genome of any bacteria, we say, okay, let's draw out the genome. Where is, you guys remember this, glucose down to pyruvate, right? That's called glycolysis, right? What's the first enzyme in the pathway? Does anyone remember? Hexokinase. Woo! Boy, fun to remember that one, isn't it? So where's the gene for hexokinase? Every organism on Earth has a hexokinase gene. Where's the gene for hexokinase? Okay, let's say hexokinase is here. Okay, and then there's an enzyme that's the next step, and that's phosphoglucose isomerase or something like that. Where's that gene found in the genome? We map that one out. Okay, that was there, and it goes this way in the genome. Right? We can map out every gene that we know in bacteria. We can look in the genome. We can look for a promoter sequence, right? We can identify where the genes are. If we do that, in bacteria, the genes are evenly spaced on the genome, like this. There are other sequences, there are other things like the origin and replication. We can find that, and we can map all this out on the bacterial chromosome. Remember, bacteria have a single circular genome, almost always a single circular genome. Okay, great. Why do we care about this? We're going to look at eukaryotic chromosomes by comparison to this. We'll find they're different in many, many ways, and that's a setup for a Zowie I'm going to get you with later in the semester. That's my little teaser, and you'll be coming back to class, right, in the future. I'm now going to introduce to you the concept of repetitive DNA sequences. What's a repetitive DNA sequence? It's a sequence that repeats. So, it is some sequence that you find many times back to back. So this A is repeated many times. And when we get to the DNA mutations and evolution stuff in this class, talk about how you get something like that. Here is a trinucleotide repeat, and we'll talk about this one also. So this one is an A, the unit is A. The unit is letter A and it's repeated. Here, the unit is CAG. And the unit is this 3 nucleotides. And then it's CAG, 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 CAG. Does that make sense? Does anyone know anything about trinucleotide repeats? Have you ever heard of human diseases associated with trinucleotide repeats?
Sickle cell?
Sickle cell anemia? No, that's a point mutation.
Okay.
Good guess. Awesome. I'm not going to tell you today. We'll learn about it another time. Okay. How do you get this thing that happens? How do you get this repeat? Just randomly by evolution? It's actually a mechanism/mistake that happens. We'll talk about that. So here is a sextanucleotide repeat. GGG-TTA. GGG-TTA. 6 nucleotides is a unit that repeats. Does anyone remember what this particular sequence was that we talked about?
Telomeres?
Yeah, telomeres. That's the sequence at the end of the telomeres, and it's repeated several hundred times. In like a stem cell or a new cell in an embryo. In me, there's very little telomere left in my cells. That's why things divide slowly. That's another discussion for cancer biology. There is, pardon me, a repetitive sequence called ALU. A-L-U. And that sequence, the ALU sequence, is about, depending who you talk to, Wikipedia said 299 base pairs. Wikipedia is wrong. It's about 350 base pairs, the original one. And that 350 base pair sequence can then be repeated. Does that make sense? So what, if this is a sextanucleotide, what is a 350? Does anyone know? I don't know. It's 349, I think is what it is. So that sequence can be repeated again and again and again. How does this happen? Anyone know? Awesome. We have a lot. Now you know some things that you will have the amazing opportunity to learn as we continue going forward in this class. It's incredible, these things we'll be talking about in the future, but totally makes sense why this all exists. So, bacterial genomes have some repetitive DNA sequences. Has anyone ever heard of this thing called Lambda phage? Yeah? What's Lambda phage? What does phage mean?
It's a virus.
Virus. Phage is a fancy way of saying virus that infects bacteria. But instead of saying virus that infects bacteria, I just say phage. Okay. It turns out that a phage, a lambda virus, that infects the bacterial cell, sometimes the genome of the virus inserts itself into the genome of the bacterial cell. So let's see, lambda phage is what? How many base pairs long is that? I'm going to guess 35,000 base pairs. Totally pulling that one out my butt here. 32,000 maybe, something like that. It turns out, in the history of E. coli, those things that live in your gut and actually you use to help you stay nourished, they break down your food for you. It turns out there was an E. coli cell, and a virus got in and infected and inserted its genome into the genome of the bacteria. And the idea for the virus is stay in the cell, and when that cell gets stressed, then the virus cuts itself out and goes away and finds some other healthy, and the bacteria dies. Before that time happened, before the virus could cut itself out and go away, the virus got mutations in it, and accumulated mutations that it became essentially a dead virus. So it was in there a long time. That appears to have happened multiple times, such that the E. coli genome has the Lambda genome with mutations in it inside of it multiple times. It is a repetitive DNA sequence of 35,000 base pairs long. Does that make sense? Okay, so what's the value of this? Well, probably that is a sequence that originally didn't have any value. It had value for the virus, not for the bacteria. And it's accumulated mutations, and maybe over time, the bacterial genome will kind of spit it out and lose it. Or maybe that's a space that now the bacteria has a little bit of DNA that can accumulate mutations and evolve into something good over time. We don't know. Come back in a couple billion years, right? Maybe 100 million years, we'll see. They may be involved in regulation of genes expression, or they may be involved in recombination. We'll talk about this more in humans later in this semester. It turns out, we think of the bacterial genome as this big, huge circle, but it's actually compacted and compartmentalized. A eukaryotic cell, the defining characteristic of eukaryotic cells is compartmentalization, right? These guys actually package their genomes, and I want you to look at this structure. If you take this, and you just grab this and pull it and pinch it in here, and then grab this, you get what we call a rosette structure. Does this make sense? And these are radial loops here. Okay? So all that needs to happen is some central protein structure needs to exist, and that protein structure just binds to different parts of the DNA and draws those together, and this big, huge thing flapping around out in the cytoplasm now becomes much more compacted and more organized. Does it make sense? Any questions so far up to this point? So this can become, oh gosh, that looks like fractal type stuff, huh, doesn't it? This can be compacted even further. You remember when I had that big piece of plastic and I showed you supercoiling? When I put a whole bunch of helical stress on one area, that area kind of twisted around itself like this, and then we got a little loop hooking up like that. We had the supercoiled area. So as gene transcription occurs in different areas, some areas become undercoiled and they supercoil backwards. In other areas where the helix gets pushed forward, those become supercoiled and they spin this direction. And so what happens is, as you add super or undercoils to this, you can actually compact the structure even farther. So it's not necessary, when we were talking about topoisomerase, the thing that cuts the DNA and relieves the superhelical twist, it's actually not necessary to release all of those. Leaving some of the superhelical twists can compact this even further in a good way. So we've talked about all this. Has anyone ever taken this drug called ciprofloxacin? It's called Cipro. Has anyone ever heard of it? You've heard of Cipro. Has anyone ever taken it? You think you've had it? So most antibiotics, so I'm on amoxicillin, and the name "cillin" at the end suggests to me it's kind of like an ampicillin type chemical. Those things attack the cell wall of the bacteria. So then my immune system can get in and destroy the strep that I had this past weekend or this past week. Most cillins are beta-lactam attackers, basically is what they are. The beta-lactam rings that screw up the cell wall. Cipro is a chemical that binds to topoisomerase. In bacteria, that enzyme is called gyrase. Such that gyrase cannot alleviate supercoiling. What was the effect of that we talked about when we were talking about gene transcription? It's screwed. Yeah. As the helical twists get pushed down, DNA gyrase in bacteria needs to be there to alleviate some of the supercoils so that that RNA polymerase can continue transcribing. If DNA gyrase is inhibited by this chemical, then the supercoils build up there, and you can't transcribe anymore. So gene transcription shuts down, and then the bacteria dies. Pretty cool. It's an alternate way of getting at the bacteria. Ciprofloxacin, I think it's called. Cipro. Any questions about bacteria before we get into eukaryotic? And in general, we'll be talking about the human genome in this context. Who cares about bacteria, right? I did on Monday. I was sick. Okay. So, the eukaryotic genome. Typical eukaryote has multiple chromosomes. The chromosomes are linear, which means they have telomeres, and they need to have the telomerase enzyme to rebuild those at various times. So, humans are the most sophisticated and advanced organisms on Earth, right? Sort of.
Depends on where you're looking at.
Sure. I mean, everything is optimized for its own environment, in theory, right? You guys are optimized for minds, right? Since we're the most complex and intelligent, we must have the largest genomes, right? 3.2 billion nucleotides. Huh? That's false. Correct. You're correct. Complexity of the organism has nothing to do with how much DNA is contained in its genome. There are frogs that have thousands of chromosomes. You can look at the number of nucleotides. So mammals have, this many nucleotides, 10 to the 9th to 10 to the 10th nucleotides in their genome. Some amphibians have 10 times as much DNA as we have in their genome. Do they have 10 times the number of genes we have? Ooh, anyone wants to answer?
I don't just like, can't really tell. There's a lot of things.
You can't tell there's a lot of things that can happen.
Well, it could be just a whole lot of dead space.
Could be a whole lot of dead space. Okay.
Or just really long genes.
What would the dead space be composed of? Introns? Based on something we've learned already today, what might be in the introns?
Repetitive sequence.
Repetitive DNA sequences. Okay. Let's go over there, said. Gold star. Who said that? Gold star.
It's on the slide.
Stole it. Stole. What? I stripped that away. Who said that?
I didn't say anything.
Yeah, there it is right there. Okay, some plants have just tons and tons and tons and tons of DNA. Okay. If we look at a eukaryotic chromosome, there are 3 essential features. And people have actually built artificial chromosomes. They've actually taken a centromere DNA sequence, because it's a known sequence. They've taken that consensus sequence, and then they've put some DNA, and then they put a telomere sequence. They put a telomere sequence, and somewhere in there, they put a sequence, and this is probably in yeast. They built an origin of replication, and they inserted that. So there's a piece of DNA that encodes for no genes. It's just a chromosome of nothing. It's just filler. And they put that into the organism, and it replicates, and it's totally fine. How cool is that? Now, what genes do you want to engineer and put on there? Put onto that chromosome. Right? So those are the really only 3 features that a eukaryotic chromosome must have. It must have a telomere. We've talked about what that structure is. It must have a kinetochore that's used during mitosis and meiosis to pull the chromosomes apart, right? And there has to be at least one origin of replication. And in humans, we call it ORI. In yeast, we call it an ARS, an ARS, autonomously replicating sequence. Genes are located in between these structures. You can't have genes in the centromere, they won't get expressed. You can't have genes in the telomere, they won't get expressed. Let's compare this. Human genome. Let's take a chromosome, let's just say it's a small chromosome, and it's the same size as this, but it's linear. Where are the genes on this chromosome? Turns out the genes will be located like this. Genes are not evenly distributed on a human chromosome. We have what we call gene deserts. And we have a gene oasis. Genes seem to be packed very closely together, and there'll be a space. And then I put one really huge gene on here, because sometimes you get these genes that are just ginormous. What was the one we talked about? Dystrophin. Many millions of base pairs long. It's spliced down to 14,000 base pairs, remember? The genome of a eukaryotic cell can contain lots and lots and lots of repetitive DNA sequences. There are things, people will get into the study of these repetitive DNA sequences and what they do and why we have them and why they exist and how they expand, how they increase in number. They break these down. There's low repetitive and highly repetitive and moderately repetitive and details we don't need to get into. Take a graduate level course if you want to learn about it. This sequence, ALU sequence, appears in the human genome about 1 million times. That sequence is 350 base pairs long. It appears 1 million times in your genome. It does not code for any gene. 350 million base pairs in your genome. So that's 10% of your genome is this sequence. What's it doing? Just hanging out?
You don't know yet either. Could do a lot of stuff.
Could do a lot of stuff, like the sand here.
Oh, it's down there. I don't know. I haven't read that yet. So, do we have any, like, actual conclusive, this is exactly what it does?
So we've found some things that repetitive DNA sequences do, and we'll get to that later. I want to have some general theoretical discussions. I want to break a mindset that you guys have on things right now. Yeah.
Would they help with like location for protein binding so that way it folds, or like so certain genes don't be expressed, like you have that combination of sequences and it's going to fold overlap each other?
What if there's a spot in the ALS sequence that a transcription factor binds to? Then that would attract, there would be a million spots in the genome that transcription factor could bind to, and you would express off of there. So maybe it's not, so ALU's a million. So what if we had some repetitive sequence that appeared 2,000 times? Now you've got 2,000 spots in the genome that will be all expressed when some certain transcription factor comes up. Is that kind of what you're getting at?
Yeah.
All right. Let me break your thinking on this. I wanna get a shift in the way you look at yourself. So you think of yourself, you're concerned about your cells, right? The smallest functional unit of life is the cell, right? General Biology 1, you learn that. General Biology 2, you learn that. So you think, okay, here's the cell, and it has all of its DNA like this, right? Metaphase cell, right? And you look at this and you say, each of these is equally important to the cell. Now I want you to go minimalist here. I want you to go in and I want you to be a piece of the DNA. And I want you to be this piece of DNA right here. How do I explain this? This piece of DNA doesn't particularly care if all of the DNA of the cell gets replicated properly. So from a survival standpoint, if you're looking just persistence through time and increase in number of copies, if the genome were to replicate, and while it replicated, it actually made 2 copies of that back to back. Okay, great, everybody else got copied. The cell has its full complement of genes, and I'm part of that cell, so I'm going to survive because everybody else got their fair due. And there's a little bit extra, there's another copy of me, and yeah, that was nucleotides that had to be added onto the chromosome. And that's a burden on the whole rest of the genome, but screw the rest of the genome. It's such a small added burden on the rest of the genome to have me duplicated that the rest of the genome will survive. But I have increased my number of copy numbers. And in selection and survival of the fittest, the thing that has the most copies is the thing that hangs around. Right? Is everyone following this? Someone want to apply a term to this. This DNA is very selfish. So the DNA in your genome is actually in a selfish battle with all of the other DNA in your genome to try to increase itself more than the rest of the genome. Does that make sense? There's an evolutionary battle in your own genome. And some are winning the battle. ALU is winning the battle. And it appears to add some advantage back to the organism at a higher level, beyond the DNA level, and those advantages we will talk about later, what those advantages are, why the ALU sequence would be so successful in replicating itself than other repetitive DNA sequences. Does that make sense? There's actually a book called Selfish DNA. It's a book you can buy and read. It's stimulating reading, I promise. Okay. So, any questions on repetitive DNA? We'll get into DNA packaging. Okay. Hopefully, in General Biology 1, you learned what a nucleosome is. Can someone define for me a nucleosome? Everybody knows what a nucleosome is?
It's the DNA wrapped around histone proteins.
DNA wrapped around histone proteins. Okay, so I'm going to define some terms for you. Histone is a type of protein. It's a protein with lots of positive charges, and it binds to DNA. You can't go to someone and say, I studied the histone gene, because there are many histone genes. It's a group. Okay? So there's, oh boy. We're going to say there are 5 histone genes, and they're repeated. You find those 5 histone genes, you might find this one 20 times and this one 30 times in different spots in the genome. They're repeated in the genome many times. And they're almost perfectly identical, almost no mutations in them. So a histone is a positively charged protein. Any idea why I'm specifying positively charged protein? Because DNA is negatively charged and this stuff interacts with DNA. And we want it to interact not with the sequence of the DNA, we want it to interact with DNA just kind of in general. And the general thing of DNA is that it's negatively charged. Okay? So there are histone, H1, H2, I gotta get this here, H1, H2A, H2B, H3, and H4. So those are the 5 histone genes. What happens is these guys get 2 of these guys, 2 of these guys, 2 of these guys, and 2 of these guys, and they form a new term, a histone octamer. It's an O, right? No, it's an A. Octamer. If 2 proteins of this guy, and 2 proteins of this guy, and 2 proteins of this guy, and 2 proteins of this guy all come together and form this kind of disc structure, those 8 proteins come together and form like a disc-shaped structure. We call that a histone octamer. And it's very positively charged, such that a DNA molecule will come and bind onto the surface and wrap around twice. Right? Well, it looks like a birthday cake. Right? So, if you have a histone octamer and a piece of DNA has wrapped around that 2 times, we call this a nucleosome. happens is this last remaining one, histone H1, comes and binds right about here. People tell you different things of what it does. What I'm going to tell you, which is not entirely true, is it grabs onto these pieces of DNA and kind of holds them together and keeps the structure intact. It just kind of sits on it, holds it there. Some people will say, this is a nucleosome, and if you take this away, it's not quite a nucleosome. I don't care. Nucleosome is when you got the DNA wrapped around a histone octamer. You can have histone H1 on there if you want, but you want to skip it, funny. Okay? Here we've illustrated this. Here's this glob of all these proteins, and the DNA wraps around it twice and comes around. And then there's a space in here. It's about 150 base pairs. And then another histone will come in, and the DNA will wrap around that twice. So if you were to take one of my cells, and you were to isolate the DNA from that one cell, it's a 2N cell, right? The skin cell that's just got a 2N copy of the genome. If you took every chromosome and lined them end to end, they're about 6 feet tall, almost as tall as me. It's a very, very thin, tiny line, right? And all of that length needs to pack into one nucleus in one cell. Just by wrapping the DNA around histones like this, you decrease that down to about 1 foot from 6 feet. You compact the length of that by 1/6, down to 1/6. Obviously, that is still too much to fit into the nucleus of a cell. So the DNA somehow has to get packaged further and further and further and compacted so it can be crammed into the nucleus of just one cell. There are a number of experiments that have run. There's probably 3 additional steps of compacting the DNA beyond this. And we don't know what they are. People are arguing what these are and trying to figure them out. It's unknown right now. So, this is a 3-dimensional model which shows the histones here and the DNA wrapped around it like this. Here they've turned it this way like this. So when you end on, there's one wrapping of DNA and there's a second wrapping of DNA. And this is just a space-going model with the DNA and the proteins shown both. The amino acid sequence. So if you've had general biology 2 with me, we talked about humans are chordates. We're deuterostomes, right? Something that's very unrelated animal, but very unrelated to us. Say a flatworm, platyhelminthes. If you took the DNA sequence for histone from platyhelminthes and lined that up for the DNA sequence for histone for humans, they're almost perfectly conserved. There is a DNA sequence, and when I say perfect, I'm going to say 70%. Maybe more, maybe 90% in this case. It says, if you mutate the protein sequence of a histone, you screw up its function. Such that the common ancestor that we had with platyhelminthes, if you branch off of that, anything that accumulated mutations along there in the histones died off. And so the histone gene has been highly, highly preserved through 500 million years. Pretty amazing. And it says what they do is very important. About half of the proteins bound to DNA are histone proteins, which means the other half are formally called non-histone proteins. Us biologists, we're really clever, aren't we? If you have DNA bound to proteins, we call that stuff chromatin. It's just no proteins, we call it naked DNA. So histones form these nucleosomes, and we're left with this 150 base pair linker region. Non-histone proteins bind to the linker region. And the reason is, these guys bind to DNA in a sequence-specific manner. The histones bind because they like negative charges. These guys bind because they like specific sequences. It may be that if you have a protein that binds to some consensus sequence, if that consensus sequence is wrapped around here, it's not available to be found. A consensus sequence can only be bound if it's in the linker region for right now, okay? Let's do that for right now. Questions? So, here's a neat experiment someone ran to show this. So, if I have my model of nucleosome, linker, nucleosome, linker, nucleosome, linker, I made this one. I took a picture and I stitched it all together. It looks really good, doesn't it? Anyway, so if I have a piece of DNA, it's wrapped in histones every so often, like this. Like this, right? There's a restriction enzyme. What you learned is restriction enzymes have a sequence they recognize that they bind to that sequence and cut it. There is one restriction enzyme. It doesn't care what sequence it binds to. If it binds to DNA, it cuts it. If I took that restriction enzyme and I threw it into a tube with this, that restriction enzyme can't bind the DNA wrapped around the histones because it's hidden. That restriction enzyme can only bind to DNA in the linker regions, right? So that restriction enzyme would cut here, and here, and here, here, and here, here, right? So the length of this DNA wrapped twice around here is about half of this linker region, half of this linker region, plus this amount of DNA. So if I had DNA and I put the restriction enzyme in there, I could cut that DNA into about 250 to 300 base pair size pieces of DNA. Right? And everything would be that, all the DNA would be that size. Let's do something a little different though. In this case, let's add the restriction enzyme instead of an hour for it to cut everything. If it needs an hour to make all those cuts, let's add the restriction enzyme and let it sit in the tube for 1 minute, and then we'll heat the tube up and destroy the enzyme. In 1 minute, that enzyme might cut here and cut here. And that's all it has time to cut. It didn't have time to find the other spots. If I took this out and ran it out on a gel, how big is this? Someone want to come up? Someone want to come up and draw data for me? Anyone daring? Nobody?
If it wasn't Monday.
If it wasn't Monday after spring break? It's okay. So this one is about 250 to 300 base pairs right here, right? So we would expect to see a band right here. This is 2 nucleosomes. This is about twice the size, right? 500 or so, 500, 600, we'd expect to see a band right there. And nothing in between, right? And this is 1, 2, this is 3 of them. I just randomly did this. So that'd be 3 times the size, right? It's about 750. Somewhere else, if I have a whole chromosome, I'll get one where there's 4 of the nucleosomes together, right? And so I will get a DNA banding pattern like that. So I'm sorry, people at home, I'm turning this off so everyone in class can see real quick. So if someone ran that exact experiment, this is the DNA that's not been treated with the restriction enzyme. And if you treat the DNA with restriction enzyme for a really short period of time, you get little bands of DNA, each one of these little bands representing 1 additional nucleotide of length of the DNA. How cool is that, huh? Experimental evidence to back this up. All right, lights are coming on, brace your eyes. That's it for today. We have lab this afternoon, and we are at the point, I've walked everyone through a PCR reaction. Nobody had a PCR reaction work, nobody at all. I'd like to meet in the conference room. I'll tell you the resources that I have available for you to work with. I am not going to tell you what to do. You need to figure out how do you solve this problem that your PCRs are not working? What could be wrong? What do you need to do to try to show what's wrong or to be successful? And I'll show you what I've got, and if you need more things that I don't have, then I will go and order those. This is big time. This is an open-ended engineering problem. You need to solve it.
What should the goal look like?
You guys, if you guys want to, as a group, to figure it out, and you each run different types of controls, have at it. We'll meet in the